Hi everyone, I'm Kentaro Suzuki from Japan, and today's my talk is about environmental factors affecting Aurelia outbreaks. So as you may know, Aurelia species look like this, and with a size up to 30 centimeter, and they distribute globally and feed on zooplankton. And Aurelia species frequently show outbreaks, and their outbreak can affect coastal food webs and damage fisheries and power plant operations. These are the typical metagenic life cycle and also the conceptual population dynamics through their life cycle. The Aurelia can greatly increase in number through both sexual and asexual reproduction, as polyps produce ephyra which further grow up into medusa. So the sexual reproduction during polyp stage has been suggested to be quite important factor of their outbreaks. Besides the mortality of planura, the mortality rate of ephyra is suggested to be quite high, like more than 80% in the first 20 days. So the interannual variations of ephyra mortality rate could also affect the intensity of their outbreaks. The possible causes of Aurelia outbreaks has been suggested, like global warming, which can boost their growth rate, and eutrophication, which can also boost growth rate and provide hypoxic condition Aurelia may prefer, and the increase of artificial substrates provide additional habitat of polyps, and the decrease of fish stock means the decrease of predators and competitors against Aurelia. The migration from adjacent areas can also increase the number of Aurelia, but most of these possible causes were estimated from laboratory experiments, and what is going on in the sea have rarely been validated. So we need quantitative analysis based on time series population dynamics. However, there are only a few time series data about the population dynamics of jellyfish because most of them are non-commercial, and there are also challenges of sampling, such as patchy distribution and net clogs. But there are only a few time series data. Those data suggested that jellyfish abundance may fluctuate with around 10-year periodicity. In Japan, there is a 9-year time series data in Ise Bay reported by Hamada 2014. And this data set is kind of unique because he reported both medusa and polyps population dynamics. So in the report, the medusa abundance, the blue points were correlated with polyp abundance in the previous winter, which produced ephyra, so orange dots. So such unique data set enables us to analyze stage-specific relationship between the abundance and environmental factors. So in this study, with the data of Hamada from Ise Bay, we aim to analyze and discuss the relationship between Aurelia abundance and environmental factors to understand the causes of the outbreaks. There are two features of this study, and the first one is to focus not only on medusa stage, but also other stages. And the second one, we treat the climate indices as environmental factors, as well as more direct variables, such as temperature, because Robinson and Graham suggested the North Atlantic oscillations and other climate indices were related to Aurelia abundance in the Gulf of Mexico. So this is, So the Ise Bay is located middle of Japan, and the bay is a semi-enclosed bay with the large river inflows at the innermost part of the bay. And the, the Hamada reported the medusa and polyp abundance from 2003 to 2011. The relative medusa abundance was estimated from the intruded amount of medusa into 6 power plants at the innermost part of the bay. And polyp density was observed in the innermost part of the bay by scuba diving in the previous winter. With these data, we estimated the survival rate after FIR release until they intrude to the power plants. So the figure shows the relationship between the polyp density in the previous winter and medusa abundance, and the dashed line is a regression line. The data above the regression line means there were more medusa than predicted from the polyp density, and vice versa. So in this study, we assumed the variations of the residuals from the regression were derived from variations of survival. We collected environmental data like river discharge, SST, SSS, and chlorophyll A. And for the climate indices, we choose Southern Oscillation, Pacific Decadal Oscillation, Pacific North American Pattern, and Arctic Oscillation as the potential environmental factors affecting their population dynamics. And with this data, we performed a simple multiple regression analysis to evaluate or estimate the environmental factor affecting polyp abundance, the survival rate, and medusa abundance. At first, I show the relationship between polyp abundance and environment. For polyps, we analyze the potential environment in previous summer to winter when polyps asexually reproduce. Among the potential environmental variables, PDO, the Pacific Decadal Oscillation, in previous summer best explains the polyp density. The PDO negatively correlated with SST, but the correlation between SST and polyps were much weaker than PDO. The PDO is reported to affect not only SST but also prey abundance. So this result can suggest that the combination of SST and prey abundance affect the asexual reproduction of polyps. Such relationship between PDO and population dynamics have been reported for other marine organisms, such as regime shift of anchovy and sardine ratio in Japanese waters. Next, survival rate after FIR release. The FIR are liberated from Aurelia in winter, and the medusa usually intrude to the power plant in early summer, so environmental factors from winter to early summer affect their survival. Excuse me. So the survival rate showed negative correlation with river discharge in January-March when the Aurelia are Ephyra or young medusa, and against river discharge in April to June when they are medusa. Compared to these two periods, the river discharge in January to June better explains the survival. High discharge causes high water exchange between in the bay and outside of the bay. So the high river discharge could flush out Aurelia out of the bay, lead to the higher mortality. Finally, we discuss about the medusa abundance. The medusa abundance is affected mainly by polyp density in the previous winter and survival rate after FIR release. So to estimate the contribution of polyps and survival on Medusa, we calculated standardized partial regression coefficient. The polyp showed higher coefficients, so that means polyp contribute more on Medusa, but the coefficient of survival rate is not so low, so the survival should be considered as well for the outbreaks. The PDO in the previous summer affected their asexual reproduction during the polyp stage, and river discharge in January to June affected their mortality after FIR release, and the combination of these two explained 80%, I think it can be pretty high, of the interannual variations of Medusa abundance in Ise Bay. So this study suggests the summer PDO affects the asexual reproduction of polyp through temperature and play, and also the river discharge in January to June affected their mortality after FIR release. And this study also suggests the importance of, of the mortality for the outbreaks, besides the well-considered asexual reproduction during polyp stage. Thank you very much, Dan Hart.